It's okay. It's okay. Thank you. Thank you so much for inviting me to speak. Oh. Thanks. um, and thank you to the organizers and Ziv for having me here. It's my second time to this meeting, and it's been wonderful. So I'm at the University of Washington and the Seattle VA, and my lab has been interested for a long time in understanding the cellular mechanisms underlying the spread of Lewy pathology in PD, and mechanisms of how GBA may be accelerating that spread of Lewy pathology in PD. And so to do that, we've used a fruit fly model, and luckily Josh Schulman earlier has convinced you that we can use flies to model GBA PD very well. So I won't go into this too much, but our fly model has a number of different phenotypes, and what we've really focused on is the accelerated protein aggregation that we see in that model. So in our GBA mutant flies, we do see increased protein aggregation that we evaluate through looking at polyubiquitinated insoluble proteins that we basically look at just from the heads. So this is the increased ubiquitinated proteins in the insoluble fraction in a Western blot. And this is just a quantification. When we express back wild-type Drosophila GBA1b just with a neuronal driver, so just in neurons, we do get a good rescue or reduction in the polyubiquitinated protein aggregates. And then we looked at this actually also with a glial driver, so expressing back wild-type GBA in our GBA mutant flies. And again, we saw a really good rescue or back down to basically normal levels of insoluble ubiquitinated proteins in our GBA mutant flies. So this really led us to wonder, to look more closely at the role of GBA deficiency in not only neurons, but also astrocytes. Another group, actually at Stanford, the Clandidin lab, had used our same flies, found the same thing. They published that a couple years ago. And then there's also been a lot of studies suggesting that astrocytes could have increased kind of rate of autophagy or ability to have a more kind of efficient endolysosomal trafficking system compared to neurons. So this was another reason we wanted to look more into the role of GBA deficiency in different cell types. So to do this, we used human iPSCs. We generated iPSCs from a Parkinson's patient who carried the GBA variant IVS2+1, which is a splice site variant leading to nonsense-mediated decay. So that variant is pretty unequivocally considered a null variant, and that was one of the reasons we chose it as opposed to other variants. And we also used CRISPR-Cas9 to generate a isogenic control, but in that process, we just by luck and chance were able to isolate a clone that was homozygous for the IVS2+1 variant. So we use that as kind of a null control. We also have iPSCs generated from an unrelated healthy age and sex matched control. And I'll also show some additional control with these control cells that were treated with the irreversible GCase inhibitors, CBE. So these iPSCs were differentiated into neural progenitor cells, and then from there we can kind of have a branch point to generate midbrain dopaminergic neurons versus astrocytes. So first we wanted to see if our GBA deficient cells have reduced GCase enzyme activity. We do see basically almost no activity or basically undetectable activity in our GBA null homozygous cells, so that was really reassuring. We see a trend towards a reduction in our heterozygous cells as well, and this is our isogenic control. And then is the protein still around in our GBA deficient cells? This is in the neurons. We do see a reduction in protein levels in our heterozygous lines, and then our homozygous line is also further reduced. This is the wild type control line treated with CBE, so of course we would not expect to see a reduction. And then we, of course, want to make sure we really did differentiate our iPSCs into different cell types, and so this is just transcriptomic profiling that is confirming that we see neuronal and actually dopaminergic, more specifically dopaminergic neuron gene expression in our neurons, and then more astrocyte specific genes in our astrocytes. So the way that we evaluated them initially for endolysosomal trafficking was by antibody staining of the cells, and we looked at, lysosomes more specifically with LAMP1, early endosomes with EEA1, and multivesicular or late endosomes or recycling endosomes with RAB11. When we stained the cells, we then looked at overall size of the stained organelles, and then individually, and then we looked at the compartment size of each organelle. So that is taking the total volume of stained organelles within a cell over the ratio of that total volume per cell of the stained organelles within the total volume of that cell. So that's what I'll refer to as the compartment size. So when we looked at early endosomes in our GBA deficient neurons, we didn't see any significant difference in the actual size of the individual early endosomes, but we did see an enlargement in the compartment size, and that was in heterozygous as well as homozygous, and then also in the control line treated with CBE. In terms of protein levels, we also see an increase in EEA1 level in the heterozygous, homozygous, and then the control line was CBE. When we looked at astrocytes, we did not see a significant increase in the compartment size. This is just a big outlier, just one single one, but otherwise there was no significant difference. And in terms of vesicle size, really no significant difference compared to the control. This was just some differences between the CBE line and the homozygous, but they were not really different from the control line. And then in terms of protein levels, again, we did not see a significant difference. There was a trend in increase in the GBA homozygous line, but it was not significantly different. So when we looked at lysosomes with LAMP1 staining, again, we didn't see significant difference in vesicle size between all of these different genotypes and the control with CBE, but we did see, not surprisingly, a pretty big increase in the compartment size of heterozygous, homozygous, and then control cells with CBE compared to the control line. And then correlation with increased LAMP1 protein as well. But in astrocytes, we actually did not really see an increase in compartment size. So here, the only one that was increased was the control line that was treated with CBE, but the heterozygous and the homozygous astrocytes did not have an increased compartment size compared to control. And then no significant difference in terms of individual kind of lysosome size. And for protein, it was a little bit unexpected, but we did see an increase in protein level for the homozygous line, and actually a decrease in the heterozygous line. And then in looking at the recycling endosomes with Rab11 staining in neurons, we do see, again, an increase in compartment size in the heterozygous line, the homozygous GBA immune line, and also the control cell line treated with CBE. Again, no major changes with vesicle size, maybe a trend, but it was not significantly different. And the protein levels were also increased for Rab11 in our GBA deficient cell line neurons, as well as the control neurons treated with CBE. But in astrocytes, we did not see an increase in compartment size and no significant difference in vesicle size either. And then in protein levels, again, Rab11 was not significantly different. This is just an outlier, not an asterisk. Yeah, in astrocytes. So we wanted to see if these endolysosomal trafficking differences between neurons and astrocytes correlated with pathogenic alpha-synuclein. And so we looked at staining of these cells with an antibody that was specific to aggregated alpha-synuclein and should not mark alpha-synuclein monomers. And what we found was that there was significant increased staining in our GBA heterozygous line, GBA homozygous line, and the control cell lines treated with CBE. And then we looked at phosphorylated serine 129 alpha-synuclein levels by Western blot in these cells. And again, this was increased in the GBA heterozygous, homozygous, and CBE-treated control cells. In astrocytes, we only saw increased aggregated alpha-synuclein staining in the homozygous line, which I think was very interesting. We didn't actually even see it in the CBE-treated control cell line. And then for phosphorylated serine 129, we did not see a significant difference compared to control cells in terms of phosphorylated alpha-synuclein levels in the astrocytes. So I know that that's a lot of things I just showed. So just to summarize, basically we see some significant differences in endolysosomal trafficking between GBA deficient neurons and astrocytes. In the early endosome, regarding early endosomes, we see an enlarged compartment size and increased EEA1 protein expression in the GBA deficient neurons, but not in the astrocytes. The EEA1 expression was only increased in astrocytes treated, control astrocytes treated with the irreversible GCase inhibitor. For the lysosomes, not surprisingly, we saw very enlarged compartment size in neurons, increased LAMP1 expression, but we actually did not really see this in our GBA1 deficient astrocytes, except for in the GBA null, our homozygous astrocytes, we did see increased LAMP1 expression. In the recycling endosomes, again, an enlarged compartment size, increased Rab11 expression, but this was not seen in our GBA1 deficient astrocytes. And these endolysosomal trafficking alterations did correlate with pathogenic alpha-synuclein, and so we saw increased aggregated alpha-synuclein and phosphorylated serine 129 in our GBA deficient neurons, but really only aggregated alpha-synuclein in the GBA homozygous astrocytes and not in the heterozygous. And then we did not see any increased phosphoserine 129 in our GBA deficient astrocytes. So we wanted to look a little bit more globally how GBA deficiency could be affecting neurons and astrocytes. We did transcriptomic profiling of them. This was a bulk RNA-seq since they were all grown in a dish separately, and And I think just initially, just looking at how looking at the differentially expressed genes for neurons versus astrocytes, there were very few actually shared differentially expressed genes between the two cell types. And actually, even when looking at the ones that were astrocyte specific versus neuron specific, the profiles were very different. So there were a lot more downregulated genes in the GBA deficient neurons compared to control neurons, a lot more upregulated genes in the GBA deficient astrocytes compared to control astrocytes. So this just initially off the bat really suggested a profound difference in how GBA's deficiency is affecting these cell types. And then taking a very bird's eye view of the key pathways that could be affected differentially between these cell types. Not surprisingly, we see genes that are involved in lipid metabolism that are differentially expressed between these two cell types, lysosomal function, ER stress. This was a little bit more of a surprise of seeing kind of neuroinflammatory genes that were downregulated in our GBA deficient neurons, and then reactive astrocyte genes upregulated in the GBA deficient astrocytes. And then just looking a little bit more closely at those lipid metabolism pathways. We see that cholesterol synthesis genes were a little bit more upregulated in our GBA deficient neurons, which I think was very interesting. Cholesterol transport, which maybe is not too much of a surprise given the function of astrocytes, but they were upregulated more in our GBA deficient astrocytes, and then fatty acid desaturation. Sorry, this is looking very faint on this, but it was more upregulated in our GBA neurons, deficient neurons. And then, not too surprising, but sphingolipid pathways were upregulated in both cell types. And then looking more closely at the genes involved in reactive, driving reactive astrocyte state. So those were pretty strikingly upregulated in our GBA deficient astrocytes compared to control astrocytes, and of course this was not the case in the GBA deficient neurons. So in conclusion, I've shown that from our model that we do have a lot of suggestion that GBA deficiency is contributing to PD pathogenesis, potentially through various cell type specific mechanisms. And GBA deficiency, many people have seen this affects endolysosomal trafficking in neurons. And this is very closely correlated with pathogenic protein aggregation. We also know GBA is affecting lipid metabolism alterations in neurons. But in astrocytes, this does not seem to be totally the case, in that GBA deficiency is not impairing endolysosomal trafficking to the same extent. It seems to be driving a more toxic reactive gliosis state in astrocytes, and our RNA-seq data is suggesting more specific effects on cholesterol transport in astrocytes. There's of course a lot of caveats to this work. iPSC-derived cells are not a perfect model. We're also looking at these cells kind of grown in isolation. We definitely want to look at them more in co-culture and more complex models. The other thing is that I've only shown you data for one GBA variant, and we'll have to see. That one was was intentionally chosen because it's a unequivocal null allele, but we'll have to see, if these results are also present in variants that are not considered a null allele, like N370S. But this is suggesting that we could potentially develop, have other mechanisms that are cell type specific to develop our target therapeutically. So I just want to finish by thanking my lab and all my collaborators. Anna Park was an amazing post-bac technician in my lab who's now going to start her PhD at Washington University. She did most of the neuronal work, and Sarah Fish, also a very talented post-bac technician, did most of the astrocyte work. I happened to have a great view of Mount Rainier when I flew over here.